Hi everyone, welcome to HM Learnings. Today's topic is apoptosis. So it is very important topic if you want to study the pathology of certain diseases like cancer and the other autoimmune disorders. So if you already know that what is apoptosis, what are the mechanisms of it, and what is the difference between the apoptosis and the necrosis, it would become easier for you to understand the pathology of the cancer and the other diseases. So let's start today's topic. So this topic will be divided under the following headlines. First, there will be a small introduction, then a definition of the apoptosis. It will be followed by the causes and the examples of it, and finally the morphological changes which occurs in the cell when the cell undergoes the apoptosis. So we know that all the cells, they start their life from a single cell called as the zygote. And this zygote will undergo then a variety of phenomena, multiple processes to finally form an complete organism or a adult. So this process in which a single cell zygote is transformed into an adult which now contains about millions of cells, or you can say infinite number of cells. So this procedure is called as the deployment. If I ask you what are the two components of the deployment, the one you will surely say that it is cell growth. So in the cell growth, there must be increase in the cell number, and the second thing is that there must be increase in the cell size. So if there is increase in the cell size and there is increase in the cell number, so the cell is growing, and if the cell is growing, the organism is developing. But what will be the another component of the deployment? So if you read the definition of normal deployment, it says that there should be death of some cells plus there should be generation of the new cells. So by this definition, we can say that the another component of the deployment should be the cell death. Now, in order to call the deployment to be normal, it should be always remembered that the rate of the cell produced should be equal to the rate of the cell die. So you can understand this definition by taking an example. Suppose we are taking an example of an organ called as the liver. So this is an organ called as liver. So like every organ, the liver also has a characteristic shape and it has a characteristic size. So suppose now the liver is only growing in size. There is only cell growth, that means there is increase in the cell number, there is increase in the cell size. So as a result, what has happened, the liver is now growing and it will definitely lost its shape and the size. So in that case, would you say that this is a normal deployment? Definitely not. It is not a normal deployment. So for that, for maintaining the shape and the size of any organ or even the organism, the cell which are produced at the rate should be equal the rate of the cell death. Okay. So now, what is the another importance of the cell death? So another thing is that you have a cell and that cell is actually infected by a certain type of virus. Now suppose this damage is a DNA damage. Now what will be the cell do? The cell know that now it is a threat to the individual, so it will decide to die. So by these two things, that the cell death is required, to maintain the size and the shape. And the second thing, it is required to prevent spread of infection. We can say a very important point, which is that the cell death is not a random process. It doesn't occur randomly. It has some purpose, and that's why the cell death is called as a programmed event, or more precisely you can say it is a programmed sequence of molecular events. That means the events are sequenced at the molecular level. So you can understand this thing by taking this thing. So there is, suppose a death signal has came. Now this death signal could be from inside the cell or could be outside the cell. This death signal will induce many molecular changes. It will trigger a sequence of the molecular changes that will finally cause the death of the cell. So this whole thing is called as the apoptosis. So now you know what is deployment, what is cell death, why cell death is equally important at cell growth, and somewhere you know that what is apoptosis. Now we look to the exact definition of apoptosis. Apoptosis is a pathway of cell death that is induced by a tightly regulated program in which cells destined to die activate intrinsic enzymes that degrade the cell's own nuclear and cytoplasmic proteins. So you can understand the definition as there is a death signal, and this death signal will trigger a sequence of the events which are tightly regulated, and it will activate finally the intrinsic enzymes, namely caspases. Now this caspase will act on the DNA to degrade the DNA as well as the cytoplasmic proteins, and it will finally cause the death of the cell. So it is called as a tightly regulated program because it is genetically regulated. Or the most frequent term which is used with the apoptosis is the programmed cell death. So it is called as a programmed cell death because it is genetically regulated. Now you know what is the definition of the apoptosis and what is deployment, why the cell death is important. So even though you forget the exact definition of apoptosis in your exam, you can write the definition in your own words. Okay, so we now talk about the another thing, what are the causes of apoptosis. Okay, so one thing I forget to mention is that apoptosis is a Greek word and it means falling away. Okay, so this could be understood as like the trees which are not required, they are falling away from the trees. Just like the cells which are not required in the body, they will undergo the process of apoptosis. So moving ahead, we talk about the causes of apoptosis. So you can also name this topic as the role of the apoptosis, or the conditions in which the apoptosis takes place, the importance of the apoptosis, and the need of the apoptosis. Okay, so the first thing is that apoptosis occurs in the two situations. Firstly, it will eliminate all the unwanted cells which now don't have any function, now they have no purpose in the body. And the another thing, if the cell is got infected, in that case also the apoptosis will take place. So what are the examples? During embryonic development, the hands and the feet grow as big. So during embryonic development, the digits of the hands and the feet, they grow as there is a tissue in between them, which is called as the web. So this web is not required in our digits. So the tissue between the digits will be undergo the apoptosis to finally shape them as what we have supposed to. Okay. The another example is the shedding of endometrium. So during every menstrual cycle, there is shedding of the layer of stratum functionalis. So that is also done by the apoptosis. Another thing, the neutrophils and the lymphocytes which are produced as a result of some kind of infection. Now there is end of the infection. The infection is not present now. So they now have no purpose. So they will undergo apoptosis. Another thing, if there is a formation of a self-reactive lymphocyte, it has to be undergo the apoptosis. Otherwise, it will trigger the autoimmune diseases. So to prevent the occurrence of the autoimmune diseases, such lymphocytes need to be died. The second importance, or you can say the second condition in which the apoptosis occurs, is injury to the cell. So the first type of injury is, suppose there is DNA damage. So if there is a DNA damage and it can't be repaired, so the cell will decide to die. Why? Because if the cell will divide, the mutations will also get amplified. So the cell doesn't want to do that. So it will commit the suicide. So DNA damage occurs when there is exposure to radiation, like in chemotherapy. Then another thing is that the anti-cancer drugs, all this will cause or trigger the process of apoptosis, so that the cancer cells die by the apoptosis and they will not cause any kind of inflammatory reaction. The another thing, or the another damage to cell, is that if there is formation of the misfolded proteins, means the proteins are formed as a result of mutated genes. So in that case, they will cause a endoplasmic reticulum stress. So that will also trigger a cell to get die. The another thing is viral infection, like the HIV. So they also trigger the process of apoptosis. So now you know what is the definition and what are the causes and the conditions in which the apoptosis takes place. Now we talk about the morphological changes which takes place in a cell when it undergoes the process of apoptosis. So here is a normal cell, and now a signal has come called as the apoptotic signal. So a signal has came, and when the signal has came, the first change in the cell is this cell shrinkage. Okay, you can see here that the size of a normal cell is more than the size of this cell. So the cell has shrinked. So if the cell is shrinked, that means there is reduction in the size of the cell. The organelles will be more tightly packed and the cytoplasm will be now more dense. After the first change, the second change will be what? The second change will be the nuclear fragmentation. Now what is nuclear fragmentation? The enzyme caspase will act on the nucleus and it will degrade it, and thus it will form small aggregates which are peripherally accumulated. Now this could be of different sizes and the shapes, and all these bodies will consist of a degraded DNA. The third change after the nuclear fragmentation is the formation of cytoplasmic blebs. So now here you can see that this is a cell membrane and it has outpouched. Okay, it has outpouched to form a cytoplasmic bleb. If you zoom out this region, you can say this is one cytoplasmic bleb. Now what does this cytoplasmic bleb consists of? It consists of the nuclear fragment, this thing, then the cytoplasm is there, the cell organelles like the mitochondria, ribosomes are also present here, and a very important point is that the cell membrane. So a very important point in the apoptosis is that even when the cell even when there is a formation of a cytoplasmic bleb, the cell membrane remains intact. Now the last change is what? The last change is finally the formation of apoptotic bodies. So now, after the outpouching or the formation of the cytoplasmic blebs, finally this whole cell will be converted into the different apoptotic bodies, and each body will have an intact cell membrane and the contents same as that of the cytoplasmic bleb. So we can again revise the changes. The first change is that there is cell shrinkage. The second change is that there is nuclear fragmentation. The third change is that there is formation of cytoplasmic bleb. And the fourth one is finally the formation of apoptotic bodies, which are then finally phagocytosed by a phagocyte. So with this we have completed the morphological changes which occur in the cell when it undergoes the apoptosis. So this is the end of this video. If you want to study this topic from the book, these are the two books you can refer for this topic. In the end, I would like to share that in the upcoming video, we will talk about both the types of mechanisms of the apoptosis, the intrinsic and the extrinsics. So if you like this video, please like, share, comment and do subscribe this channel for more such videos. Till then, keep learning.